So I'll just quickly introduce her. And I think she's from Triple IT, so most of you know Anubha in many different ways. So the most prominent role that she took up recently was the Dean of Academics, and now she is focused more on research. But yeah, so a quick formal introduction of Anubha is she did her BTech in Electronics and Communications Engineering from University of Delhi, PhD from IIT Delhi, and then she did a master's, on education from University of Maryland, College Park in the US, and then she was in many leadership roles. I guess she was the assistant director with the Ministry of Information and Broadcasting, Government of India. That's very interesting. Like, so she has, experience in uh, high level managements in government, which is interesting. And then also a very deep interest in research and a lot of work she has done the past few years. She has been a faculty of IIIT Hyderabad, NSUT Delhi, and, of course, IIIT. She's a professor in the electronics department, and she works on a lot of things which are not which are deep into bioinformatics and biology. And she works on cancer genomics, cancer imaging, biomedical signal and image processing, including fMRI, MRI, EEG, ECG, signal processing, and wavelet and deep learning. And she's a senior member of IEEE Signal Processing Society, right? And a member of IEEE Women in Engineering Society. She's an associate editor of IEEE Access Journal, IEEE SBS Magazine, e-newsletter, Frontiers in Neuroscience, and IET Journal of Research. She is also a member of IEEE SBS Committee of Women in Signal Processing and chair IEEE SBS Delhi section for Jan 2019 to December 2021. With that, I welcome Anubha for her talk, which is, and the title of the talk is Datasets and Tools, released by SBILab. That's how she calls her lab. Easy to remember because SBI is one of the major banks. Sorry, so datasets. So that is the title of the talk. Over to you, Anubha. It's all yours.
Thank you, Debarka. Thank you so much for a nice introduction. So I'm sharing my screen. Just let me know if it is visible.
Yeah, please.
So I don't know. This seems to be. So
it's yet to come, Anubha.
yeah. Just give me a second. It's not allowing me to share.
Is it?
Yeah.
Well, Shifra.
So shall I send you my PPT or?
No, you should have the.
All panelists have all the rights. Please try one more time sharing your screen, ma'am.
Okay. Because it has never become a problem ever before.
Okay.
Showing me some weird things when I'm asking.
Why don't you make Anubha the host for the moment?
No, just a minute. Let me put my laptop on. Sorry, this is a mess, but
I've also shared the hosting rights with Dr. Anubha.
so should I try again?
Please try one more time.
I've also given her share screen right to Dr. Anubha.
So there is this option share screen. It is asking me whiteboard, iPhone. It is asking me weird things to actually put on. Okay. Yeah. Hello. Good morning, everyone. So this is Anubha Gupta, and I'm working as professor in Department of ECE, and my lab is SBILab. So given the topic of this workshop, so I decided to deliver a talk on data sets and tools that have been released by my lab. So as Dr. Debarka also mentioned, we have been working in a number of areas in biomedical signal and image processing. So I'll be giving you a brief recap of the tools and the data sets and the codes that we have released. If you have any question, you can also stop me in the middle because it is a brief presentation. So the research in my lab is actually largely in the area of cancer imaging, that is microscopic medical image analysis, cancer genomics, cancer survival analysis. So these three threads are the main threads in which a lot of work is going on. Besides this, there is one very active thread on which I'm working since past 1 year, which is on cardiovascular disorders, on ECG signal processing that we are working with some of the hospitals in Delhi, GB Pant Hospital, Mamsi, etc. So a lot of research has also happened in the area of neuroscience, and a lot of my students have worked on this topic, and I've also been working with some national and international groups on this. So one of my PhD students, for example, did her PhD in fMRI signal processing. EEG signal processing, also one of the students graduated recently from IIT Delhi, and a number of other students are also working in the similar areas. Signal processing for communication engineering, because I'm largely a signal processing person, but I love to apply signal processing methods or evolved methods for the application areas. And of course, research and education is my deep interest. Not a very active thread. I keep writing a little bit in this area as of now. So this is my research group at SBILab. Shiv is my student who largely worked on cancer imaging and published a lot. He has recently joined a job. Akanksha, Vivek are working in cancer genomics. Ashutosh is working on diffusion tensor imaging, that is again neuroscience area. Sachin has joined in cancer imaging. Shubham worked on cancer imaging only. Ekansh and Lakshya, they worked on EEG signal processing. So my collaborators, I'm largely collaborating with the lab oncology unit at IRCH. Professor Ritu Gupta is heading that. And her team consists of Dr. Gurvinder, Dr. Jaina. Shruti was a JRF on one of the cancer imaging project and really helped a lot. At IIIT Delhi also, I'm collaborating with two colleagues with Dr. K. Shriram and Dr. Gaurav Ahuja in computational biology department. So I've divided my talk into four short sections and would like to discuss what all we have done so far. So the first is, so this is the largest thread that or I would say in past 7 years, I have really spent a lot of time and energy on this particular thing with the hope that sooner or later we will be able to release some affordable, low cost, AI-enabled blood cancer detection tools. So this is in blood cancer imaging, and we have worked on B-type ALL and multiple myeloma. Both of these are actually white blood cancers. And the idea behind the whole project is that the existing laboratory techniques like microscopy, flow cytometry, or molecular studies on bone marrow aspirate, they are really expensive and require expertise. So we wanted to develop some affordable low-cost tools that can be deployed easily in any pathology laboratory. So the steps involved in this process are that we have to collect, first actually recruit subjects who are suffering with cancer and some who are healthy, then collect microscopic image data. So generally the slides are prepared from either the peripheral blood smear or the bone marrow aspirate, and then the images are captured. Once they are captured, then comes the process of identifying how many number of cells of a particular type are actually cancer vis-a-vis healthy, and then looking at the ratio, one can determine what is the stage of the cancer. So, the in-between steps are actually tricky. For example, if you look at these 3 slides, they have slightly different shade at the background, etc. So most of the computer vision techniques that employ, for example, here we would require to do cell segmentation and then cell classification. So most of these techniques are actually, the results get altered if there is a color variation. So hence we need to do, so if you see these 3 different slides, they have slight color variations across them. So we need to do a stain color normalization. We say that it is owing to the staining process, which is done from time to time at different points of time with different bottles of this chemical, and hence lead to slight variations on the slides. So, so the process includes stain color normalization, then cell segmentation. For example, if you can see my cursor, these are the cells of interest. And these become tricky when it is actually a slide which is prepared from bone marrow aspirate, because at that time, lots of cells are actually clustered together. For example, if you look at this. So there are a number of challenges even in cell segmentation, because at times the color of the cytoplasm is very similar to the background, or the 2 cells are touching and they can be touching at each other at maybe by the nucleus or by the cytoplasm, where the boundary might be very, very thin. So all these challenges lead it to a very difficult problem when it comes to, particularly in multiple myeloma, where this amount of cytoplasm is sufficient. Whereas in BALL, because they are the immature blasts, it's largely the nucleus. So we can just extract the nucleus and then try to do healthy versus cancer classification. So, but post stain color normalization and segmentation, it comes the challenges of the classification of healthy versus cancer cell. And why the problem is difficult? Because even under the microscope, pathologists say that morphologically the 2 cells appear very similar. So when we had made this problem around 6 years back, at that time, the doctor's intuition was that it cannot be done, because with the eye, we cannot determine even under the microscope whether the 2 cells are different. But we moved ahead and we have been able to find that yes, with the help of computer vision, that is the deep learning or the machine learning methods, we are able to really discriminate between healthy and the cancer cells. So we conducted in the recent past 2 medical image challenges in prestigious IEEE conference, which is IEEE ISB in 2019. As well as in 2021. So this conference is actually the flagship medical image conference of IEEE Signal Processing Society. So the most recent challenge was called SEC-PC. This challenge was conducted, so it actually, the challenge runs for about 4 months, and it concluded in April as part of the conference, where there was 1 day when we were given half a day to conduct the session. And the task was cell image segmentation from microscopic images of multiple myeloma, the one that I just showed, that this is a difficult problem in for segmentation. We released this dataset publicly at IEEE Dataport and Kaggle, and the data is available. It is also mentioned clearly at our lab page of SBILab under resources. The leaderboard is also open, which is here. So if anyone wants to download the data and run on this data and devise their own methods of cell segmentation, they can do so, and there is a test data for which there is no ground truth labeling or annotation available. So they can check their results on the test data by uploading on the leaderboard, which is active. So another challenge that we conducted was in 2019. Again, this data is also available, and this statement is wrong. The data is available at TCIA, the Cancer Imaging Archive, and this data consists of around 15,000 cell images which are carefully curated and annotated. So let me tell you, all these data sets we have collected, for example, BL dataset over a period of 2 years, and we did a lot of meticulous collection as well as annotation, separated them at the subject level, that is no two subjects data is being mixed up here. These cell images are actually, after we have done stain color normalization, cell segmentation, then curated and then release this data set. Again, the test data is also available, but the ground truth of test data is not available openly, but the leaderboard is open and anyone can actually check the results on the leaderboard of whatever method that they have devised. So for the first challenge, the global teams participated in this. So for the first challenge, the highest score that was in terms of F1 score, and that is around some 93%. And this is after the team had applied a number of ensembling of number of deep learning methods which are already available. So these are the state-of-the-art segmentation methods, and they did ensembling of that. And after that, they have been able to achieve an F1 score of 93%. So you can imagine how critical or how difficult the data set is. So if you are interested, then you can explore these data sets as well as participate in the challenges. So these are the 5 data sets that have been released so far by my lab. So the first one I just mentioned, CNMC data set, that is on the Cancer Imaging Archive, free downloadable. And so this is on, these cell images are from actually BALL cancer, and hence this is on classification of healthy versus cancer cells. So the data is around 118 subjects and well separated at the subject level. So then the other data set, which is MIMM data set, we released in 2019. So this is also on the cell segmentation of multiple myeloma, the smaller data, but very neat and very carefully annotated data. The third data set is again interesting data set. It has 30 images of big slide images of around 1920 into 2536 size, each image size of BALL cancer and multiple myeloma cancer, and it is largely for stain color normalization in case you want to develop your own methodology. And every image is very, very different. You can see the color contrast because we collected each of the data set over a period of 2 years, and hence a lot of variation went into the data which was collected from one time point to another. So the fourth one is related to the recent imaging challenge, which is I just described. So it is available at IEEE Dataport as well as on Kaggle. And the fifth data set, this is actually in EG, and this data set is corresponding to 11 subjects, 7 subjects who are healthy and 7 who are suffering with intellectual and development disorder. So this data we collected, we worked with Tamanna NGO for some time, so they have subjects who are suffering with IDD, that is intellectual and developmental disorder. And so this data is collected through a Emotive Plus sort of a headset, and we also got the 3D this headset printed and made them familiar with that, and only then we could collect this data. So while they're listening to music or in the resting state, so we have collected this data to find out whether there is a sort of a different brain networks that we can infer, or are they altered in healthy controls vis-a-vis IDD subjects. So that's this data set, and this is also freely available, as well as we also released a pre-processing pipeline of the EEG data, which is collected from the emotive sort of a device, so which is also available freely for anyone to download and use that pipeline. So these are the codes that we have released related to the cancer imaging. So stain color normalization, the codes are available here. You can download the codes, and this paper has complete details of the method. The second one is classification of cancer versus healthy cells. So we also devised our own deep learning pipeline, and so far this is sort of working best as far as leaderboard is concerned. So it's around 95% F1 score that we are getting on this. And the code of this deep learning architecture is also available for free download at this site. So then I come to the next work that we have done, although we have some more papers in pipeline, but I would present one that has been recently accepted. So one of my PhD student, Akanksha, she has worked also on this cancer risk staging. So we came up with a AI supported modified risk staging for multiple myeloma. And here, so the idea was that we wanted to, so far we have two risk staging systems. One is the ISS and the second one is the revised ISS. So these are sort of standard risk staging systems. RISS came somewhere around in 2015. So while ISS uses only 2 parameters, RISS also uses the genetic features. And these genetic genomic tests, they are not easy or are not economical in all regions of, for example, developing countries like India. So RISS definitely works better than ISS, but we wanted to achieve similar or better performance than RISS without using the genetic features, so that it really works out in our settings in given our laboratories and all the medical infrastructure setup. So we used 6 parameters: age, hemoglobin, calcium, beta-2m, albumin, and EGFR ratio. And we devised our strategy. We built it on the AIMS data, but we validated on the publicly available MMRF dataset. And we found that our design strategy or the risk staging that we developed is really working much better than ISS and is also comparable, and at places it is working better than RISS. So the calculator is available online here. And so this is the whole design strategy that we have AIMS data. So we know that not every subject has all the features. Sometimes some of the values are missing. So we imputed missing data only in the training set, not on the test set, but we imputed missing data using one of our own devised methodology, for which also the codes have been released. You can find lots of codes on our lab page under the publications. With the paper, we are also giving the links of the codes. So then we did a test train split, and we used k-adaptive partitioning to find the newer threshold. So this is also important, that we found that not the existing thresholds on the features or the parameters which define whether the albumin greater than this or less than this, whether this is the normal range or this goes into the abnormal range. So we determined our own thresholds. And then we applied this univariate Cox proportional hazard model, and then we assigned weights to different features, and then calculated weighted score and computed then the adjacency matrix, did the clustering on that. And once we had the clusters, because clustering is unsupervised, so then we used train J48 tree to assign the labels. And then we looked at the entire pipeline, how it worked and whether it did a better job or not compared to the existing ISS or RISS. So we labeled this strategy as the MRS, Modified Risk Staging Strategy for subjects. And we validated on the MMRF dataset. So this is the calculator, and it is also available on the lab page that I mentioned above. If you click on this, then this calculator is available. So for example, this is the decision tree finally that came out, and we got it verified from the doctor also, whether whatever are the rules that we are getting, are they really making sense? And here you can actually reset and put the entries. Anyone can put the entries and figure out what is the risk stage. So the lowest risk stage is 1 and the highest risk stage is 3. So it is very easy to use calculator and uses only 6 parameters which can be easily measured also. So then I'm going to tell briefly about cancer genomics work. So our work is largely, most of the work is actually under review, has not gone through with publications, but we are hopeful that very soon we'll be having some papers published in this area. So one significant work that we have done is on developing a new bioinformatics pipeline for novel miRNA detection. We have named it as MIRPipe. So we know, for example, miRNAs are key regulators of cellular processes. So we explored a number of pipelines, but we found that somewhere there were a large number of false positives, etc. So we developed our own end-to-end computational pipeline that works on RNA sequencing data. And our workflow actually detects unique novel miRNAs. It is also able to find functionally same miRNAs, that is paralogs, that is having the same seed and the region other than the seed region, but is actually located at different genomes, at different position in the genome, and miRNAs which are belonging to the same phylogenetic tree, that is having the same seed regions. We validated and compared the performance of this MIRPipe with the available most recent state-of-the-art pipelines. And for this, we used synthetic as well as cancer dataset. So this was interesting because in the real dataset, we know that the ground truth is not available. So every time we were struggling with the thing that how do we know that if this method has found so many novel miRNA and our method has found so many, so how to compare the performance? So finally, we decided to build our own synthetic tool as well. So we call it as MIRSIM tool. This is available again for free download, and is we have the complete, this tool is available at Zenodo, and anyone can use this tool. And it's an interesting tool because it generates the synthetic FASTQ and FASTA format files for RNA sequencing. So we upfront decide, for example, how we can add the sequence, we can create the number of reads, and we can then, in those reads, we can add known miRNAs, known piRNAs. We also altered a bit in the sequences of miRNAs and piRNAs. So we added them as the other sequences that should be rejected and should not get identified as miRNAs as well as piRNAs. Then we also added some sequences which are actually have been detected as novel miRNAs by some of the recent papers, but they have not been yet annotated and available in the standard databases like Mirbase or PyRNA databases, etc. So a user can choose how much percentage or which all sequences they want to add, and they can generate this. But with the expression counts also, you can decide how many reads you want to put for a particular mRNA sequence, etc. But then once you generate this synthetic file, so that means you know the ground truth, and now you can validate different pipelines. So we validated our pipeline. For example, I'm just showing you the results on 1 million read depth generated using MERSIN tool. And these are the results on known miRNAs on the left-hand side. So if you see, so our pipeline has some portion of it has been imbibed from MIRDEEP2 pipeline, which is also free, but we modified it considerably so that our false positive reduce. So if you look at the comparison of MIRDEEP2 and MIRPipe, we find that the accuracy has increased. So even MIRDEEP2 is doing a good job when it comes to accuracy. But when it comes to the F1 score, that is how many false positive, false negative, etc., it is dealing with, its F1 score is very low, whereas the MIRPipe has a very good F1 score also. So it gains both in accuracy as well as F1 score. And these are the other pipelines which are around. Some of them are actually machine learning based algorithms like Mirage and SNRA toolbox. So they're not able to compete with these pipelines to a very large extent. So if you look at the performance on whether on known miRNAs, novel miRNAs, we also included the workflow for piRNAs in the same pipeline. And we find that overall, when it comes to the performance, it really surpasses all the pipelines by a huge margin. So this pipeline will, as soon as our paper goes through, will actually, we have created a Dockerized version of this, and we'll be releasing this pipeline as soon as this work is also through. So a little bit recap of what, at least related to the data set. So I already said that we collected EEG data set of individuals with intellectual and development disorder and healthy controls under rest and music stimuli. So Krishna Veni and Blessing were actually at the Tamanna NGO, and they were the special caretaker in whose presence, who actually collected the data for us. And this is the Emotive one hit set. And this is the complete pipeline. So this pipeline is also, because Emotive is actually headset. So the sensors here are unlike the traditional EEG data capturing devices. These sensors are actually dry sensors. So they're not pasted. They don't fix on the head. So there is a slight motion. So that is why the SNR, or the signal to noise ratio with the data collected with this Emotive, is not of very high quality as compared to the other data set, other devices. So it really requires a very robust pre-processing. So we developed this fully automated pipeline. So many pipelines are available, but generally they are not fully automated. And you need to develop some insight about, for example, artifact rejection, how to do that, etc. And it takes a while. So we developed this automated pipeline, which is very good, and you can just download and use it on any of your Emotive collected data as well. It is available at Mandalay along with the data that we have released freely. So many students, UG, PG students, research interns have contributed to our work. I'm working with lots of faculty, doctors around within Delhi, and also with some people abroad as well. And so far, so the data that have been collected and released, at least in cancer genomic and the cancer imaging, we have got grants from Department of Science and Technology, as well as Ministry of MITI, which is now called MITI. We also thank the Infosys Center for AI and Center of Excellence in Healthcare for time-to-time support that they give us. IBM was technical co-sponsor for our IEEE ISB 2019 challenge. Springer actually consented to publish the challenge proceedings that came out as a book. And last but not the least, I would say we really owe a lot to our staff at IIIT Delhi. They are extremely supportive, and it really makes the environment so conducive for us to carry out our research. So anyone interested, please visit my lab. If you want to collaborate with me, you're most welcome. Do contact me. And any question, I would be happy to take the questions. Related to any of the work, I can go in detail, but I was told by Professor Rava that perhaps I need to be talking about the tools and data sets released. Yeah, so I stop here.
Thank you, Anubha. I think